there in terms of HbF levels, the clinical correlates in terms of being VOC-free, and all the side effects are consistent with busulfan-related conditioning across these patients. So we're quite bullish about how the trial's moving forward. We obviously did a streamlining of our operating model with Vertex earlier this year, which allows for us to think about a coordinated global launch once we get there, and also a coordinated global manufacturing scale up to meet the needs of patients that are suffering from these terrible diseases.
Got it. That's really helpful. So it sounds like for EHA, you're not going to say how many more patients you're going to have there, but is it fair to say that there will be some additional patients worth of data?
Well, I mean, the typical way we do this, we have a great deal of discipline around how we look at the data and how often we open the database. And I think we only want to do it if it's absolutely called for and necessary. I think we also, at the same time, operate with a sense of transparency. We want to show the data that we have in the interest, not just in the interest of investors, but mainly because we want all the investigators to know what's happening, the patients to know what's happening, and that's how we're going to gain momentum in the trials, but also establish our preeminence in the field.
Got it. Okay. And from the abstracts, it sounds like one of the takeaways is that you're seeing consistency across the different genotypes, and that shows the breadth of the approach.
Yeah, I think severalfold. Consistency across genotypes, the consistency at which the fetal hemoglobin goes up. It's not a one-off. You don't see the variability that you see with gene therapies, and that's one of the things that if you think about what the FDA and other regulators care about, one of the biggest concerns they have is variability patient to patient. And you've seen that with both AAV-mediated therapies, gene therapies, as well as lentiviral-based gene therapies. And here what you're seeing is the patient-to-patient variability is kind of limited. You're seeing similar consistency in the elevation of fetal hemoglobin, similar kinetics, and that similar pattern is leading to similar clinical outcomes. And I think those are all very good facts to support the notion that CTX001 is applicable across all transfusion-dependent thalassemia patients that are severe, and across all sickle cell patients that have severe disease.
Got it. Okay. And will you say if you're going to be providing additional details on editing efficiency, bone marrow engraftment, et cetera, at EHA?
Yeah, again, we don't comment on the nature of data that you may see. I think we always balance the notion of being very transparent about our data with having the right forum for doing it. I think you want to have these are various things that we're looking at in the trials, right? We measure all sorts of correlates. We measure all clinical and non-clinical aspects, including bone marrow editing, the peripheral editing, the percentage F cells, et cetera. And we want to package the data appropriately so we can interpret the data correctly. And that's the balancing factor between being, completely disclosing these data, but making sure they're robust. And we will show data that makes sense in this context as we get to a greater number of patients overall in the study.
Got it. I thought you were going to disclose the data at the Jefferies conference. Well, so let's see. So based on the initial data you've shared with regulators, I guess what's been the feedback, and are there any specific points of negotiation?
I think the discussions with regulators are going really well. I think, again, these are not one-off meetings. This is a continuous set of meetings we have with the regulators on account of the fact that we have RMAT designation in the US and PRIME designation in Europe. And both of those designations came on account of the fact that the regulators think this is highly promising therapy. And the discussions are going really well. I think the two aspects typically of discussions around how you get to a filing package are around clinical and non-clinical, and the non-clinical mainly being around CMC aspects. So I think on the clinical aspects, there is, as the data matures, the question becomes, what is the bar in terms of how many patients you need, and what is the the amount of follow-up you need on each of these patients, or the last patient. That's what counts, right? Last patient in plus the follow-up time to get to a data set that could be filing ready. And then on the CMC aspects, the question is, as you scale up and mature and diversify and globalize your supply chain and your supply at manufacturing and supply, are they all comparable? Are they all sufficiently characterized and qualified to meet the needs of patients in a commercial setting? And I think that's the discussion. I think those are the two aspects. One could be rate limiting versus the other, but at this point, I think we're trying to bring all these to converge at the same time point as we continue to mature our data and discuss these aspects with the regulators.
Got it. Okay. And you've mentioned potential for 15 to 20 patients per trial, so maybe 30 to 40 patients total, and duration of follow-up of 12 to 18 months to be sufficient. Can you talk about how you're getting to those projections, and do you have some alignment with regulators on this already?
We haven't provided any guidance saying this is officially aligned with regulators or anything like that. I think this was our ingoing hypothesis as CRISPR Therapeutics, saying this is what you may need from a regulatory standpoint, given the data we're seeing in the early patients, and if the data set hold across all the patients, right? And I think you've seen conditional approval for Bluebird in Europe based on a similar number of patients, and you're seeing now others attempting to file with small number of patients, given curative type of data. And so it's very reasonable to think that 15 to 20 patients, if all of them or almost all of them end up being transfusion independent or VOC free, that could give us a lot of confidence that this is ready for wider access in society. Now, the specifics, of these will have to be discussed with the regulators, because it's different for a safety database versus the efficacy population. It's not the same type of statistics you're doing, because here, you're seeing a majority of the patients have the effect, or almost everyone having the effect is the expectation. In which case, how do you really think about statistics in the classical sense, where typically we've had drugs that only give you a response for a certain part of the population, and the effect size is not as clear. Here, it's black and white in terms of effect size. So again, it's a bit of a discussion with regulators, judgment call, and ultimately we'll get to alignment around that.
Have they provided any specifics on what the safety database should look like?
Again, so we don't discuss these regulatory discussions. Any specific regulatory. We'll combine all of it and give you an update at some point this year.
Okay.
Regulatory discussions, but I'll say is the regulators have been very supportive, both in the U.S. and globally.
Guys, quick note on this, really important, right? We're talking about regulators. We know how the FDA needs to approve CTX001. So these are really good questions. Pay attention to these types of questions, guys. All right.
That update, right? I think if you look at the range of all the analysts on Wall Street, there's a range out there, and you yourself have put a specific timeline, put a potential timeline on it. I think these discussions will be much easier, and provide greater clarity once we've got to that clarity with the regulators. I think once we feel like we have enough confidence that we're all aligned around what that filing package is and what that might take, I think we'll provide that update ourselves and say this is what we expect. I think before that, it's a bit premature to speculate on what the timeline might be for a launch, et cetera.
Got it. And maybe last question, you mentioned at the beginning of the year, you had the meeting with Vertex and discussed some of the operational changes you could make. And you've also been ramping up the CMC and manufacturing capabilities. I guess, what's the goal for manufacturing, and what is it going to look like commercially?
Well, I think we certainly want to be ready for hundreds of patients, but eventually get to thousands of patients. I mean, that's the goal. I think If you look at what Vertex did in CF and the global reach, the notion of saying we're going to cure every cystic fibrosis patient and get to all of them around the world, that was a bold ambition. And I think a lot of the interest in sickle cell is it's a very analogous disease with known molecular genetics, same patient, the burden that the patients have to deal with, and we have a potentially curative therapy. And so the ultimate ambition and goal here is to get to every patient across the globe. Now, the realities of the costs and the complexities of cell-based therapy at this point, autologous cell-based therapy at this point, means that we need to start with Western Europe and U.S. and Western Europe initially to get to all these patients. But eventually we want to keep scaling up. I mean, this will be a linear process, as again, with our goal being that supply capacity is always greater than what the demand is out there.
Got it. Okay. And maybe shifting gears to your off-the-shelf CAR T programs. So you've commented on providing new data from 3 separate off-the-shelf CAR T programs, including 110, and initial data from 120, which is BCMA, and then 130, which is your CD70 this year. Are you providing more granularity on timing at this point?
We have not provided granularity on timing, but I think for 110 and 120, they're both heme malignancies. Generally, our sort of MO has been that we target, the initial data release. We're going to do it as a company release because the data are early. But eventually we want to get to scientific medical meetings, because the objective function is to get all the PIs excited about the trial, get them to be fully knowledgeable about the data, and have that discourse in a medical forum about what the data mean and the implications of it. And so that's generally been our philosophy around data. With 120 and 130, we haven't put a timing out there around the data disclosure. For 110, I think as the data continue to mature, we'll figure out what the right forum is for that data disclosure.
Got it. Okay. And just to clarify, it sounds like for 120 and 130, since it'll be initial data from both of those programs, it'll likely be in a press release?
Very likely a company press release or company presentation, although we haven't ruled out that we could, do it at a conference
okay,
as well. So I think we're still making that decision. But generally, I think our philosophy has been company release for the first data release.
Got it. Okay. And let's see, for your cell product strategy, maybe if you can talk a little bit about that. And one of the big questions with off-the-shelf CAR Ts is getting to the maximum level of T cell expansion and thinking about persistence as well. And so how does your 80-20 mix of cells play into that question?
Yeah, at a very high level, I think we have a few companies playing in the allogeneic space. With CD19 CAR T in particular, you have ourselves, Allogene, and Precision, all playing in having different strategies for persistence, right? I think all of us, in effect, have the same strategy for targeting. You insert a CD19 CAR. We do it with CRISPR. Others do it with Lenti, and then we all knock out the TCR locus to make the cells not have so we prevent graft versus host disease. But then for persistence of the cells and to enable the cells to be around long enough to kill the cancer, we use different strategies. Our strategy is to knock out the MHC class I complex by knocking out the beta-2M gene. Allogene obviously have used CD52 knockout with the alemtuzumab-like antibody to ensure persistence. That comes with a greater conditioning burden upfront, and then Precision have a different strategy. Now, I think we still think beta-2M is an important part of making these cells stealth. We've actually incorporated beta-2M into our Regen Med program as well. And I think, our strategy where we have 80% of cells knocked out with beta-2M knockout versus the ones that have beta-2M intact allow us to understand the kinetics in patients and stratify them by response to see what's actually happening with these cells, right? So we're going to learn a lot about that, especially, in fact, all the new edits we introduce, we're going to create some heterogeneity in the cell population mix, because the heterogeneity plays to our advantage, because we'll learn what the cells do with and without the edits in the same human being, the same patient, which is the best controlled experiment. And we'll know that data before others do, and be beneficiaries of learning what the contribution of each edit is that allows us to build on our products over time. So I think, is there NK cell killing with beta-2M knockout? Yes, there is some. And we saw that even in preclinical studies, but not every cell was killed by NK cells. And I think we'll in our initial data release for CTX110, recall that we saw cells out to 180 days in one of the patients that was tracked that far in terms of cell persistence. The other thing that's happened in the meantime, by the way, if you think about your conference last year versus this year, the focus was so much on persistence and how long cells last, and what the expansion of cells are. Now, if you look at some of the filing packages for auto CAR Ts, this durable CRs are not all correlated to. There's some correlation to expansion, but it's not such a clear correlation in the sense that you have greater expansion leads to durable CRs. It's not the case. There are lots of durable CRs with low expansion, and it's also not clear that greater persistence lead to more durable responses as measured by B-cell aplasia. There are patients that have B cells come back very quickly, and they're still durable responders, and patients that don't have it and have relapsed. So it's a very complex equation. I think there's the durability that you're ultimately aiming for is a function of how hard you hit the cancers up front and some sort of surveilling function. And how hard you hit the cancer up front is not just simply a function of expansion, but there's more to it. I think all these things we're understanding as we go along in our trials. And I think if you see durable, even 6-month durable responses with allogeneic CAR T, because you won't have the benefit of following patients too long, 6-9 months, with single dose or single administration in a reasonable fraction of patients, that opens the door for a product with allogeneic CAR T. And once allo CAR T is in, you will just see it move to frontline settings much, much faster than autologous CAR T settings. And in fact, it may dominate autologous CAR T because in CD19, because the data may be equivalent, but so much more convenient and fit with the system.
Got it. That's helpful perspective. And for one of your programs with the CD70, so CTX130, there's a lot of interest in that one because it's differentiated. You're focusing on solid tumors there. I guess, what are you going to be looking for in the initial data to reinforce the approach in solid tumors? What would
so they're talking about the CAR T programs, right? CTX 110, CTX 120, and CTX 130.
If you see a CR or any response in solid tumors with a CAR T, allogeneic CAR T, would be a dramatic advance for the field from a scientific standpoint, because so far we haven't seen responses with CAR Ts and autologous CAR Ts in solid tumors, right? I mean, there have been a number of trials, whether it's prostate or other indications, there hasn't been a response. So any response is good from a scientific standpoint, but as a company, we want to make a drug, and a drug that's going to be very competitive in the field. So in renal cell carcinoma, for instance, with third line therapies, after you've exhausted the PD-1s and the TKIs or CTLA-4, in third line therapy, there's really nothing. They don't have any options, and these patients have very short overall survival. I think any response there would be a welcome. Would be very welcome from that investigator community and from physicians treating renal cell carcinoma. And I think we want to understand other aspects of how CAR-Ts work in solid tumors too, along with responses. We want to see what the kinetics are. We want to see if there's exhaustion. We want to see how this, cell expansion behaves in solid tumors versus heme malignancies. There's a lot to learn from it, but we're quite bullish about CTX130 because we see a lot of CD70 expressed on these cells in renal cell carcinoma, and it's a very highly immunologic tumor. But I think, as we get to higher dose levels and the right dose levels, we'll start looking at the data and provide an update at some point this year.
Yeah, I like how he
If
said
we'd
he's
be the
bullish
first
about
company
130, the
to show any
CTX130.
sort of activity in solid tumors, the CAR-T, and that's just the start of the field, right? It's only going to get better over time.
Right. And Sam, we're almost out of time, but I wanted to check on the Regen Med and your collaboration with BioSite. So you mentioned prior that there are regulatory meetings in the US and the EU ongoing. Any key takes from these meetings, and is everything on track to get the phase 1/2 started in 2021?
Yeah, we remain on track with together with our partner BioSite. I think the regulators are getting much more comfortable with iPS derived cells or embryonic stem cells, and just the notion of stem cells in general as a product. I think you've seen a few other INDs in this space recently. And I think we've done a lot of work to characterize these cells and fully understand them, given the nature of the disease and the patient population we're dealing with. So we remain on track, and we'll provide further updates on what the actual product is. We've disclosed some of the edits in our product, but not all of them. There is a number of edits in these cells, almost 5 to 6 edits. So it's a very sophisticated cell construct, and that's something that's enabled by a powerful CRISPR platform. It's not doable with other platforms, and we continue to advance.
Got it. Interesting. Maybe last quick question. So you mentioned for the Regen Med, for that program, you're going to use the B2M knockout for that. If it's inside of the pouch that could be used, would that avoid the NK cell detection?
It's likely. I mean, I think, we do want the device and the cells, that area, to vascularize, because that's how these cells are going to get nutrients to survive for a long time. And so whether we can block out NK cells, we don't know. But I think there are other ways to avoid NK cells. I think the beauty of using stem cells is they can make as many edits as you want with the CRISPR technology and make them much more stealth than you can in the CAR-T context with the first generation products. So I think that's something that we'll see, how much threat is there from NK cells, and we'll learn as we go along.
Got it. And to close out, what should investors be focused on for the rest of the year for CRISPR?
Well, I think, needless to say, the most important area of focus this year is the immuno-oncology readouts for 110, 120, and 130. I think, but if you look at where we stand now, this gives us tremendous upside with limited downside on these CAR-Ts. But beyond that, I think, getting to the clinic with a diabetes product and making further progress with our in vivo programs are all things we are looking forward to as a company, to make us not just the leading player from a timeline perspective, but also from a capabilities and diversity perspective of programs. We'll have iPSDR programs, in vivo programs, and everything else as well.
Excellent. Okay. Thanks so much for joining us today, Sam. It was great seeing you.
Yeah. Thank you, Maury.
Take care.
All right, guys. So hopefully you guys appreciated this. This was Jefferies healthcare conference, virtual conference. It happened today, and this was live. And this was between Dr. Sam, the CEO of CRISPR Therapeutics, and Mr. Raycroft here, who was asking good questions, especially the questions with ViaCyte towards the end, which is type 1 diabetes that they're tackling, which we talked about in this channel, that will enable type 2 diabetes in the long term if this program gets FDA approved. So great news so far, nothing negative. I mean, nothing really new, but it's always refreshing to see the CEO be bullish. That's his words. He's very bullish about CTX 130, for example. So it's really interesting to see some of these words, some of these keywords used by Dr. Sam. So hopefully you guys appreciated this. Like this video, subscribe, and let's see each other in the next video. Thank you very much.